हाँ। Good morning.
I, Pawan Pujari,
And I, Divya Jadhan.
are delighted to welcome you all to a very special and newly introduced segment of the 6th Student Research Congress 2025, the Hackathon Oral Solids category. This year marks a significant milestone as we expand our scientific horizon with this dynamic, innovation-driven hackathon. It is an initiative proudly curated by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by the University of Mumbai and supported by our valued industry collaborators, ACG Worldwide and SciTech Center. The hackathon is not just a competition, it is a creative battleground where science meets speed, innovation meets practicality, and ideas turn into real time solutions.
To set the tone, let me share a thought that resonates perfectly with today's session. Every solid dosage form begins as an idea, compressed with science, shaped by innovation, and delivered with purpose. This simple yet profound message embodies the spirit of hackathon: find, innovate, transform. In the Oral Solids Hackathon, participants step beyond the theoretical boundaries. They engage in rapid problem solving, formulation strategy, real challenges, and practical innovation.
To all the teams participating today, your presence reflects your courage to challenge norms, your readiness to experiment boldly, and your determination to redefine what is possible in the world of oral solid dosage forms. May this hackathon ignite your creativity, sharpen your problem solving skills, and inspire breakthroughs that shape the future of pharmaceutical industries. We wish all participants the best. Let innovation lead the way, and may the best idea win.
We are honored to be joined by such distinguished experts. Their guidance and experience greatly enrich today's proceedings. It is my privilege to introduce our esteemed judge for this session, Dr. Parizad Elchidana. Dr. Elchidana is a distinguished leader in the global pharmaceutical industry with over 35 years of experience spanning research, development, quality systems, and advanced regulatory sciences.
She holds a PhD in pharmaceutical sciences along with a postgraduate specialization in pharmaceutical technology from the prestigious State University of Ghent, Belgium. She is also a trained internal auditor for ISO 9000 quality management systems and holds additional diplomas in business management and computer science. Throughout her illustrious career, she has contributed extensively to generic drug development, especially in patent non-infringing products, QP-driven innovation, and AI-enabled process development.
She has successfully delivered numerous FTF, 505(b)(2), and NCE-1 submissions and has several patents and research publications to her credit. She has played a pivotal role in setting up and managing greenfield pharmaceutical projects for global investors and has even led complete US FDA remediation programs, culminating in successful facility clearances. Dr. Elchidana has served as the Managing Director of Apotex Research Private Limited, the Indian arm of Canada's leading generic pharmaceutical company. Later, she was invited to Dubai as the Chief Technical Officer at Pharmex Pharmaceutical FZ LLC, where she oversaw facility establishment, qualifications, and regulatory approvals. Currently, she serves as the Principal Technical Consultant Pharma at ACG, bringing her deep expertise to support innovation and excellence in the industry. Alongside her industrial accomplishments, she has remained deeply connected to academia as an honorary visiting faculty, technical advisor, recognized PhD guide, and a highly respected examiner for MPharm and PhD programs. She is also an invited speaker at numerous national and international conferences. She has also contributed as the president of the Controlled Research Society Indian chapter and co-chair of the AAPS Western India chapter Mumbai discussion group. We are truly honored to have such an accomplished professional with us.
Thank you. Thank you, Dr. Parizad. Thank you for being here.
We also have with us Dr. Shubhashish Chakraborty, General Manager of Global Product Management at ACG Capsules. Dr. Chakraborty is an accomplished pharmaceutical professional with a PhD in pharmaceutical sciences from IIT BSU and over 17 years of diverse experience across leading multinational companies. His career spans senior roles in R&D, business development, sales, and global product management, reflecting both scientific depth and strategic relationships. In his current role at ACG Capsules, he leads global initiatives in new capsule technologies, driving innovation from early development all the way to commercialization for both pharmaceutical and nutraceutical applications.
Prior to this, Dr. Chakraborty contributed significantly to Evonik India Private Limited, where he held prestigious leadership positions, including Regional Sales Director, Director of Key Account Management, Head of Business Development and Contracts, and Head of Technical Services for the Asia South region. His work focused on supporting major pharmaceutical companies in developing complex injectable systems using advanced biodegradable polymers. He began his scientific journey at Lupin Research Park, working as a formulation development scientist, laying the foundation for a career marked by excellence and innovation. Dr. Chakraborty has an impressive academic footprint with over 40 international publications, 2 book chapters, and numerous recognitions. His achievements include consecutive publication scholarship awards from IIT BHU Alumni Association in 2009 and 2010, and the Best Alumni Award for his extensive professional accomplishment.
Beyond his scientific contributions, he is also a passionate author, having written 3 self-help books, Happiness Decoded, Room to Grave, and Touching the Core, all of which are globally available, including on Amazon. Above all, Dr. Chakraborty is deeply committed to student growth. He invests significant time as a coach, mentor, and career counselor, inspiring young minds across the country. We are indeed honored by his presence today. His vast experience and expert perspective greatly enrich our deliberations and lend significant value to this session.
Dr. Rajesh S. Parab, Honorary Treasurer of International Pharmaceutical Excipients Council of India. Dr. Parab is a distinguished professional in the pharmaceutical domain, widely respected for his leadership, technical expertise, and deep commitment to advancing in excipient sciences in India. In his role at IPEC India, he plays a pivotal role in strengthening industry standards, promoting quality, and fostering collaboration between academia, regulators, and the pharmaceutical industry. Known for his clarity of thoughts, practical insights, and progressive approach, Dr. Parab has been instrumental in guiding discussion, shaping policies, and supporting scientific excellence across various national platforms. His contributions have been made a meaningful impact on both research and practice in the field. We are truly honored to have him here with us today. His presence and perspective add immense value to our proceeding. Please join me in warmly welcoming Dr. Rajesh S. Parab.
Very welcome to you, sir. Dr. Rajesh, Dr. Subhashish, and Dr. Parizad. Your presence will definitely add a lot of value to this solid oral segment. And I'm sure being a pioneer, ACG, being a pioneer, plus all 3 of you have a wonderful experience, would definitely make a difference. So
Thank you.
So over to you. Yeah.
Before we begin, I request all participants to carefully go through the instructions displayed on the slide. The presenters have to rename themselves as per the allotted quotes if not already done. The presenters are requested to stay online, switch on their camera when their code is called out. Pre-recorded video will be presented on the screen. Participants cannot reveal their name or any affiliations. Each participant is requested to stay alert for their roll call. All the participants should be present for their session. All members from the group are allowed to answer during the Q&A session. The duration for the Q&A session is limited to 5 minutes.
With the permission of our esteemed guests, we are ready to begin the oral presentation competition for Hackathon, Oral Solids category. Our participants and audience are eager to experience the innovative and impactful ideas presented today. Also, I kindly request the judges to confirm that they have received the scorecard.
Received. Yes,
Yeah,
received.
we've received. Yes.
I would call upon code numbers OS004 to switch on your camera. We will be presenting your video now.
Hello, ma'am. A very good morning. Am I visible and audible?
Yes. Please proceed.
Okay.
A very good morning to everyone. My code is OS004, presenting my innovation thermal alert packaging, a novel visual alert packaging system for thermal stability of oral solid medicines. Proper storage of pharmaceutical formulations, it is very important. If the formulation is not stored properly in desired temperature limits, the formulation may get degraded or lose its effectiveness. Also, the oral solid formulations like tablet, capsules should be strictly stored at the room temperature around 25 to 30 degrees Celsius. And other formulations like vaccine, biological insulins, they need strict cold storage condition in refrigerator around 2 to 8 degrees Celsius. If the formulation is exposed to this above temperature limits, the drug may get degraded or lose its effectiveness. And in country like India, there is continuously fluctuation in temperature. In weather like summer, the temperature sometimes goes above 40 to 45 degrees Celsius. And this temperature is not good for pharmaceutical medicine storage. So, also, according to the Arrhenius theory and rule of butane, as the temperature increases by the 10%, the drug degradation rate increases by that twice. So, this is a very serious problem that the medicine should be strictly stored as the desired temperature limit. Also, to check the seriousness of this problem, we went to pharmacy shops. We conducted a little survey in the chemist shops, talked with pharmacists, asked them questions like if they stored their pharmaceutical formulation in proper temperature, and is there any effect of the temperature on the pharmaceutical formulations. Also, we asked them, is there currently any solution available to solve this problem? So the 100% pharmacists told us that the pharmaceutical formulation like medicine should be stored at the proper temperature, and if it is not stored, the drug may get degraded. And there is currently no any system available to tell us the pharmaceutical formulation is properly stored or not. Here are some my photos with pharmacists while I have conducted the survey. So to solve this problem, presenting my innovation thermoalert packaging. A thermoalert packaging, it is a color changing solution to this problem. Thermoalert packaging is the innovative packaging system which is simple, low cost, lightweight, user friendly. It offers real time temperature monitoring. Means thermoalert packaging will show the formulation is properly stored or not through a visible color change. Thermoalert packaging offers us two types of packaging. One is thermoalert reversible packaging, and the second one is thermoalert irreversible packaging. Thermoalert reversible packaging, it is specifically designed for the formulation like tablet, capsule, and other formulation which should be stored at the room temperature around 25 to 30 degrees Celsius. And thermoalert irreversible packaging, it is designed for the pharmaceutical formulations which should be stored at in refrigerator around 2 to 8 degrees Celsius. Material required are thermochromic pigments, different polymers like PVA and PEG, and PVP, and some resins. Here I have attached an image of thermoalert reversible packaging. Here, my innovation. I have made three types of innovation. First one is thermoalert reversible packaging. Second is thermoalert irreversible packaging, and third one is thermoalert indicator scale. Thermoalert reversible packaging, it is applied on the secondary packaging of the pharmaceutical formulations like tablet, capsules, and others. It is in dark blue color or purple color, and as the temperature increases to 30 degrees Celsius, it will change its color and it will go to the sky blue color. Dark blue to the sky blue when the temperature goes above 30 degrees Celsius, and the temperature goes back to below 30 degrees Celsius, it will revert back to the dark blue color, which we can storage our medicine properly by just seeing it that the color, by seeing the color of the pharmaceutical packaging. The second one is thermoalert irreversible packaging. It is designed for the thermolabile formulation. This packaging is stable in refrigerator, and as the formulation is exposed to higher temperature around 30 degrees Celsius, for continuous 10 to 15 minutes. The formulation, the packaging will form a permanent stain on the pharmaceutical formulation. So we will see that stain and we will get the idea that this formulation is exposed to higher temperature. So this is not safe to use. The third one is thermoalert indicator scale. Thermoalert indicator, this will be applied to the pharmacy shop's shelf. So we can monitor the temperature of the pharmacy shop shelf by looking just its color. So this will alert the shops uh, pharmacy shop staff that about the temperature fluctuations so they can store their medicine properly. Uniqueness. This is first of its kind packaging, means there is no currently available pharmaceutical packaging in market like my packaging that will show the medicine is properly stored or not through a visible color change specifically. This is non-toxic, low cost. That's why it is scalable. This is smart, simple, and user friendly. And this is designed for all type of pharmaceutical formulations. And another innovative approach we have added to this is QR enabled layer. Upon scanning this QR code, it will play an educational video for the educational or informative video for the user. By seeing that video, the user will get all the information about colors. Means which color means gives us which indication. Like purple means it is properly stored. Sky blue means we need to store it properly. What does mean that permanent stain? The stain indicate us what this type of indication it is given in that video. Video is available in both the languages Hindi and English. If you have some time, I request you to please scan this QR code through Google Lens and you will see that video. I have made that video, so you get all the information about the how the user will get information. These are the methodologies. For reversible packaging, we need to select the thermochromic pigment, then we give acid treatment to it, then we process it with different polymer matrices, then we cast and coat it on the secondary packaging. The second one is thermoalert irreversible packaging. To make the thermoalert irreversible packaging, we need to take a base layer of paper, then we apply a polymeric PEG layer on it. We took two different grade of PEG, PEG 400 and PEG 6000. We melted them down and got our desired PEG, which is specifically get melt on the 30 degrees Celsius upon continuous exposure to 10 to 15 minutes. Then we dry and settle it with pigment layer on it. These are some results. First one is thermoalert reversible packaging. Reversible packaging, it is initially in purple or dark blue color, and as the temperature goes to the 30 degrees Celsius, it will change its color to the sky blue. And when temperature goes back to the below 30 degrees Celsius, it will revert to dark blue color. And the second one is irreversible. This is how it will look initially in refrigerator, and as the continuous exposure to higher temperature 10 to 15 minutes, it will develop a permanent state like this. Currently, the irreversible packaging is still under development, but the test batches shown a satisfactory result, and we are improving it to handle mechanical shocks while transportation. So the thermoalert packaging shows a immediate and clear color change when temperature goes to the 30 degrees Celsius, and it is confirmed repeatability, and trial batches of irreversible patch showed a good result. This packaging is stable in moisture as well, non-moisture sensitive, and gives good repeatability of result. And it is low cost and materials are non-toxic. That's why it is shows good scalability. Here I have added a demonstration video. In that video, I will show you how my thermoalert packaging changes its color. So we have developed a thermoalert evaluation chamber to show the packaging, how this packaging change color. This thermoalert evaluation chamber is attached with a heat source. We have used this heat source and the thermometer to show the current temperature of thermoalert evaluation chamber. So we are putting this thermoalert reversible packaging into our thermoalert evaluation chamber. So this is my thermoalert indicator, which is currently in purple color because currently this temperature is below 30 degrees Celsius. I have put this into my evaluation chamber. Now I will apply some heat to it and we will see the packaging, how it changes its color. So currently it is completely showing purple color, and I am applying some temperature to it, and we are seeing that the packaging is changing color. See, the packaging is continuously changed to a sky blue color from dark blue, and temperature is above 30 degrees Celsius specifically. So this sky blue color will revert back to the purple color within a minute when the temperature, the formulation is properly stored. So this is why we call this reversible thermoalert packaging.
invite the judges to ask their questions to their respective presenters.
Hello, sir. Very good morning.
Yeah, good morning and thank you for the lovely presentation, which was also accompanied by video demonstration. It's an excellent idea. I have a couple of questions. One
Yes,
is
ma'am.
for your reversible packaging. You said it has to be put on the secondary pack. Now, in
Yes, ma'am.
India, if you see, most of the tablets and capsules are being sold with the primary pack itself. So, how do you plan to handle those cases? And my second question is on the irreversible one, where like in refrigeration, if you are talking of, it'll be mainly for parenteral products. And parenteral products would be either in ampoules, vials, pre-filled syringes. How do you plan to handle those?
Okay, ma'am. So the first question, you said that the secondary packaging. I have applied it to secondary packaging, so it won't make contact with directly with the formulation, but it is completely non-toxic and safe. So we can apply it to primary packaging as well. But as a student, I haven't technologies to attach it directly on blister package, but it is completely stable. So in future, we can attach it to blister patch pack of the pharmaceutical products, so we can directly monitor the temperature of the storage temperature of the pharmaceutical formulation. The second one is irreversible. It is applicable to the vaccines, peptides, and some injectables parenteral dosage form, and it is stable in refrigerator. And as the formulation goes to exposed to higher temperature for 10 to 15 minutes, it will develop a permanent stain. And this is how we will get to know that this formulation is exposed to higher temperature. Hello?
Yeah, thank you.
Hello,
Thank
am
you
I
for
out?
that. Yes,
Yes, ma'am.
thanks.
I have a question. I think this looks like a very nice idea. Has there any
Thank you.
work carried out before this or similar to this? Because this relates to a change in color because of subjecting it to thermal, exposure. So, any kind of related work which has been done in this field?
So, sir, currently there is no such big work available in market. I have done literature survey as well. In food technologies, there is a little work, but not that much. In pharmaceutical, there is no currently work. Specifically, we use thermochromic pigments. They change color on temperature changes. So I have taken them and processed with polymer and developed this new innovative packaging for solid orals and pharmaceutical products specifically. So this is completely unique currently. No, as such, more work on this topic.
Okay, and when you say that this can go with the packaging material, is there any specific packaging material where it can be compatible or it goes with all?
It goes with all. Sir, the pigment which I wear, it is very rugged and it can be applied with different polymers. Actually, I have tried this with PVP, PVA and different resins. All the pharmaceutical packaging shown a good result and repeated result. It is compatible.
And have you applied for any patent on this or publication?
No, sir. I am currently a master student. This is my M.Pharm project and I am doing it. It is yet to complete it. That irreversible packaging, we are developing it to get the best version of our packaging. So after that, we will take all the patents.
Okay, thank you.
Yes, sir. Thank you so much.
Hi. Hi, excellent idea. And especially in country like India, where the temperatures vary so drastically. As you said,
Yes,
more
sir.
than 45 degrees it may reach, and very important
Yes,
for sensitive formulations to stay within the given shelf life so that they are effective. You have made it very simple so that it can be applied on a secondary pack or primary packing. Have you done any studies on the robustness once it is applied? Is it a scratch resistance? So will it come out easily with a touch of a finger?
No, sir. I have applied some coating on it of different polymer coating, so that coating will prevent it on touching, and it is moisture sensitive. Completely, it is give good glossy look if we are taking it in hand hour. So this is completely stable.
Okay, nice. Thank you.
Thank you very much, sir. Thank you so much.
Any further questions from the judges?
No, not
No,
for
we're
me.
done.
No, I think
Okay,
so.
thank you so much. We go ahead with the next presentation.
Okay.
Thank you,
Okay,
judges,
sir.
for your
Can I leave, ma'am?
valuable questions. Yes, you can leave.
Okay, thank you so
Thank you,
much. Thank you.
judges, for your valuable questions.
For subsequent presentation, code number OS006, kindly switch on your camera. We will be presenting your video now. Thank you.
Hi everyone, a very good afternoon. So today my topic for the oral solids hackathon presentation is NanoMacCap for cancer therapy using functional excipient coenzyme Q10. So here you can see a diagrammatic representation of the monoclonal antibodies that are trapped in coenzyme Q10 matrix, further on leading to the nanoparticles formation and further on to capsule. So coming to the background, rationale and problem statement, breast cancer, which is one of the common cancer that is seen among the women, and it is also one of the leading cancer at the second position globally. So the background is like breast cancer remains a leading cause of cancer mortality, and the current therapies such as the monoclonal antibodies as well as the conventional nanoparticles, they are facing limitations such as the poor tumor targeting, systemic toxicity, instability, rapid clearance, and suboptimal intracellular delivery, which often exhibit low drug loading efficiency and limited therapeutic performance. So our rationale is we are here using the coenzyme Q10, which is a lipophilic antioxidant, which also serves as a functional nanoparticle matrix, which has the antioxidant properties, which reduces the oxidative stress and allows the controlled drug release, which is nothing but a monoclonal antibody. Here it is margetuximab, and which further on improves the stability, enhances the tumor targeting, and as well as reduces the toxicity. For example, the toxicity of cardiotoxicity, which is caused by one of the monoclonal antibody that is used for the treatment of breast cancer, trastuzumab. So here it has the potential to reduce the toxicity or the side effects that are caused by the other monoclonal antibody therapies. So here the problem statement is the conventional nanoparticle carriers as well as the current delivery systems that lacks the high loading capacity, tumor specificity, and optimal therapeutic efficacy. So here, with our novel and the innovative formulation, where we are using the coenzyme Q10 as a functional excipient, we are focusing on these limitations to get reduced. So here you can see the referenced articles at the below. Coming to the next slide. So here the chemicals required, here the drug is margetuximab, which is a Margenza, which is a brand name, and the nanocarrier type is coenzyme Q10 nanoparticles, and the excipients used PVA and various buffers, and the target is HER2 protein. Here you can see a diagrammatic representation. In case of normal cell, here the cells grow and divide normally, and whereas in case of HER2 positive cancer cell, the cells grow and divide faster. And the target here is HER2 protein, which is the human epidermal growth factor receptor 2. And the method which we are going to formulate the nanoparticles is double emulsion solvent evaporation technique. And here you can see the diagrammatic representation where the monoclonal antibody is acting on the target of HER2 breast cancer cells. The references, the articles which we have referred at the footnotes. And coming to the methodology, here you can see the methodology. The antibody, which is the margetuximab, is taken in a buffer and then further emulsified, where we are using the coenzyme Q10 as a matrix and vertex sonication, and the primary emulsion is formed, and then the primary emulsion is added into PVA and further on sonication, and the secondary emulsion is formed, further on solvent evaporation, washing, lyophilization, freeze drying, and then further on storage. Here you can see margetuximab, which is the monoclonal antibody, where they are loaded into the coenzyme Q10 matrix, which is the novel and the unique idea of this formulation, and the stabilizer solution, high shear, higher pressure, and the emulsions are formed, primary emulsion and the secondary emulsion, followed by the evaporation, solvent evaporation, freeze drying, powder, and the capsule. And the article references at the footnotes. Once the formulation is done, then we are going to focus on the kinetics, in vitro release kinetic. Here you can see the methodology that we are going to follow. And this is a methodology which we have cited from the comparison references. And the results we are going to estimate is whether the antibody release occurs in a sustained manner or not. You can see the article references at the footnotes. And coming to the characterization methods, the first one is dynamic light scattering, through which we are going to estimate the size and the polydispersity index, whether we need to confirm whether there is a uniform particle size distribution or not. Coming to the encapsulation efficiency and the drug loading, here we are going to estimate that it's going to be encapsulation efficiency is going to be estimating to be 50%, as well as antibody loading, we are going to estimate it to be 50%. Article references at the footnotes we can observe where we have taken the information as a reference. Coming to the immunoblot, Western blot, here we are going to check whether the released antibody is strongly binding to the cancer cells or not. And coming to the next methodology, immunofluorescence. Here we are going to see whether the clear cellular binding of the released antibody and the proper intracellular staining. And we are also going to do the other characterization methods such as the FTIR, UV, HPLC, LCMS, and so on. And the article references which we have cited the information at the footnotes. And coming to the conclusion, here and data analysis before the conclusion, we are at the TRL2 where we are focusing on the technology concept and the application formulated. We are focusing on the formulation by using coenzyme Q10 as a matrix material, where the monoclonal antibody margetuximab, for example, it can be trastuzumab or pertuzumab as well. They are loaded into the coenzyme Q10 matrix, where it is acting as a functional excipient. It acts as antioxidant property, as well as it also reduces the side effects caused by the monoclonal antibody therapy. And we are expecting it to reach the TRL5, where we are going to demonstrate its performance in the relevant biological environment. So overall, this progression will ensure that formulation is scientifically validated, functionally optimized, and it is ready for the preclinical pathway development. So our conclusion is like, there are very limited articles or the references are reports where the nanoparticles encapsulating monoclonal antibodies, and there is no coenzyme Q10 used as a matrix material for delivering the monoclonal antibodies that has been documented till date. So these positions our NanoMap cap as a unique and the innovative nanotherapeutic platform, which further on enhances or enables the efficient encapsulation, controlled release, enhanced anti-tumor activity, but this formulation is remain untested. So here coenzyme Q10 acts as a multifunctional matrix, which acts as an antioxidant property, as well as it also reduces the adverse side effects caused by the monoclonal antibody therapy. Here the antibody, we are estimating it should remain functional after encapsulation, and it should protect the antibody and enable intracellular delivery, and should enable targeting intracellular cancer proteins which are previously inaccessible to MABs, and the articles that we have referred at the footnotes. And we are also focusing on the dual targeting, which is EPR effect, where the monoclonal antibody nanoparticles should passively accumulate inside the tumors with the help of this through the EPR effect. And once inside the tumor, the map should recognize and bind specifically to the particular cancer cell antigen. So by this EPR dual effect, which is EPR passive targeting and active targeting map antigen binding, will approach and increases the drug concentration in the tumor, which also reduces or lessens or minimizes the exposure to the healthy tissues. So here you can see the pictorial representation of the EPR effect given below. So we are going to conclude that our formulation is a novel and innovative, where we are using the coenzyme Q10 as a matrix. So by this, we are expecting high loading efficiency, stability, as well as the therapeutic efficiency. So thank you for giving this wonderful.
I now invite the judges to ask a question to the respective presenter.
Good morning, everyone. Is my voice audible?
Yeah, we can hear you. Thank you for this presentation, and I'm a bit confused. Coenzyme Q10 is an active. So how did you decide to use it as a functional excipient? And
Actually, there are many references, ma'am, actually, where they have used as a PLGA, polymeric nanoparticles. So we have came to a conclusion that coenzyme Q10, which is acting as an antioxidant, lipophilic antioxidant, it also has the properties of reducing the side effects that is caused by the present monoclonal antibody therapies. So we are
And
using
this-
this coenzyme Q10 as a matrix material instead of the polymeric matrix.
Right, but it in itself produces the active effect. But my second question to you is, what is the quantity of coenzyme Q10 that you will be using per dose that you will administer of the active?
This is an idea, ma'am, where we have at the starting stage where we have thought like we should use coenzyme Q10 as a matrix material. We didn't enter into the formulation part yet.
Okay, because I mean, keep in mind the daily dose that
Yes.
coenzyme Q10 can be given, and there are chances that you may exceed that
Yeah,
when you are making the formulation.
our idea is like along with the monoclonal antibody, we also want to reduce its side effects along with the functional excipient coenzyme Q10.
Right. And also keep in mind the stability, because I've not seen any stability study done. So
Yes.
yeah, wish you all the best. I think lots of work still needs to go in.
Yes, yes.
Thank you.
Thank you.
So quick, a caution here about using the term functional excipient. So this is not an official term. Excipient means which doesn't have any therapeutic impact. And when you say functional, then it doesn't lead to anything. You know, when you are trying to connect it with excipient. So I do not know if you have any other thought process behind it, but I think as Madam also mentioned that this has got its own activity. And then calling it as an excipient, that may result landing up in a controversy or a debate.
Okay, sir. We are using it as an excipient, but we also estimating the results of functionalities like.
So once it's functional, it's no more an excipient. I mean, if it has got an active.
Okay, sir. We have put forward an idea towards it.
Sure.
Hi, it's a very good idea to reduce the side effects of anti-cancer drugs. But this is a domain which needs a lot of scientific pre-research and post-research, because you're entering into an area which will demand approval from regulators. So clinical trials, as Madam rightly said, the dose has to be matched. Your active dose is 250 mg typically used, and coenzyme Q10 daily dose is 100 mg. So if you are trying to use it as a matrix, I don't know whether the 100 mg is sufficient enough to overcome that. So a lot of literature work. No, I know there is a very limited out there. This will be required, and then getting into it's currently it's a theory and a thought. So to show that it's really working. Also, there are literatures which says antioxidants typically hinder the effectiveness of anti-cancer drugs, because they would sometimes release the free ions which will work on the cancerous cells to kill them. And if you have an antioxidant, it may reduce the effectiveness of that. So please look into that matter.
Okay, sure.
Thank you.
Thank you.
For your valuable questions. Dear judges, as we have not received any video from code OS009, we will skip that and move forward. Code number OS010, kindly switch on your camera. We will be presenting your video now. Thank you.
Hello, welcome to this presentation. In this video, I will presenting the research work corresponding to the code provided. The presentation will cover the background and rationale of the study, methodology used, the key result obtained, the data analysis performed, and the major conclusion drawn from the work. Let us begin with the first slide. Parkinson's disease is a chronic progressive neurodegenerative disorder caused mainly by the loss of dopamine-producing neurons in the substantia nigra. This leads to tremors, rigidity, slow movement, and balance problems. The condition was first described by James Parkinson in 1817 in his essay, An Essay on the Shaking Palsy. One of the effective treatment approaches that includes MAO inhibitors, which increases dopamine availability in the brain and help improve motor activity. Safinamide is a selective and reversible MAO-B inhibitor. It helps reduce off episodes and improves motor activity when used along with levodopa, especially in patients who show limited benefit from other treatments. The rationale of this research is to enhance the manufacturing feasibility of safinamide tablets using direct compression and to improve drug dispersibility by incorporating novel co-processed super disintegrants such as FMELD and Crayon T314. The goal is to develop a patient-centric orally disintegrating tablet with rapid disintegration, faster onset of action, pleasant mouthfeel, and uniform dosing. Using quality by design principles, we aim to identify critical quality attributes and optimize the formulation to ensure robustness, reproducibility, and reduce wastage of materials. The aim of this study is to design and optimize patient-centric safinamide, orally disintegrating tablets by use co-processed super disintegrants and a mixed spectral design of experiment approach. The objective include evaluating drug excipient compatibility using FTIR and DSC, studying the physiochemical properties of the excipients and preparing preliminary formulation to select the most suitable super disintegrant. Further objectives include applying quality by design for formulation, optimization, and assessing in vitro performance through flow properties, hardness, friability, disintegration, resolution, and content uniformity. In this study, several systematic steps were followed to develop safinamide orally disintegrating tablets. First, compatibility studies were carried out using FTIR to identify any possible interactions between safinamide and the selected excipient. FTIR spectra were recorded to confirm chemical stability and excipient suitability. Next, a mixed factor design of experiments was used to set different levels of super disintegrants and evaluate their influence on key responses such as hardness and disintegration time. Formulation were then prepared by direct compression using optimized ratios of disintegrated diluents, sweeteners, and lubricants. Finally, the tablets were evaluated for appearance, hardness, friability, waiting time, drug content, disintegration, dissolution, and overall T314 in varying proportion. Safinamide was kept constant at 50 mg across all batches, while super disintegrated levels were systematically varied according to the DOE design. These trials help identify the most suitable combination that could produce rapid disintegration, good mouthfeel, and adequate mechanical strength. Results and discussions. Their results indicate the safinamide was compatible with all selected excipients with no major interaction seen in FTI studies. DOE modeling showed that higher level of FMELD combined with moderate level of Crayon T314 produced the more desirable outcomes. The optimized formulation achieved a disintegration time of less than 30 seconds, a hardness range of 3 to 3.5 kilograms per square centimeter, and nearly 90% drug release within 12 seconds. Based on similar ODT studies, the formulation is accepted to offer good test masking, low friability, and uniform drug content. Overall, these results suggest strong potential for developing a patient-friendly ODT suitable for dysphagic patients with Parkinson's disease. This slide summarizes the disintegration performance and dissolution behavior of the trial formulation. Since safinamide ODTs are intended to rapid dispersion, disintegration time played a major role in the selection of the optimized batch. Among all formulations, levels of FMELD and Crayon T314 showed the fastest disintegration, some completing with just a few seconds. The disintegrates can achieve rapid dispersion, improved convenience, and potentially better treatment adherence. Although this is a conceptual formulation based on theoretical modeling, the approach demonstrates promising potential for improving oral drug administration, comfort, and compliance in Parkinson's therapy. This aligns with overall goal of developing easy-to-use, patient-friendly medication system. These references support the theoretical background, formulation principles, choice of excipients, evaluation method, and standard scientific explanation of ODTs. Our presentation is based on established pharmaceutical literature. Thank you.
I now invite the judges to ask their questions to their respective presenters.
Thank you for the presentation. Was really interesting. Which software did you use for your design of experiments? Hello. You're on mute. Can you put your mic on? Yeah.
Yes, ma'am. Are we audible?
Yes. Yes.
What was the question?
Ma'am, you repeat the question once again?
You done your experimentation using design of experiments. So I just wanted to know which software did you use for that?
Software. Ma'am, it is based on a theoretical.
Okay. Let's not waste time on that. Did you work out the design space for the formulation based on whatever critical quality parameters that you had identified?
No, ma'am.
You have not worked on that. Just one last question or a point. Here, the disintegration and dissolution is very, very critical. You are talking about 12 seconds, but your slide shows 12 minutes. So just keep that in mind. On a couple of slides, I show 12 minutes instead of 12 seconds. Yeah?
Okay, ma'am.
Right.
No questions.
Yeah, hi. It's a good idea because Parkinson's patients sometimes are non-cooperative. It may help them. Now, if you give something like this. I have seen in your presentation you have mentioned FML, Cryon-T. These are the brand names or trade names. So it will be good in a presentation if you use the scientific name or the chemical names of these excipients that you're using. And also to check the compatibility. Have you done any stability of this, or it's just a theoretical model?
It is a theoretical model, sir.
Okay, because for oral disintegration time, the disintegration may increase if it is not kept properly or there's incompatibility of the excipients.
Yes, sir.
Okay, thank you.
Thank you, sir.
So I just have one question is, are there two presenters here?
Four, sir.
There are 3 co-presenters and
Okay.
Yeah, no more questions from me.
Thank you, judges, for your valuable questions. For subsequent presentations, code number OS011, kindly switch on your camera. We will be presenting your video now. Thank you.
Good afternoon, respected judges and fellow presenters. Today, I am a research-based innovation under the theme Innovations in Oral Solid Dosage Form. Our innovation is based on design and development of patient-friendly buccal strip of varicalin tartrate with taste masking and mucoadhesive properties. Varicalin tartrate is a nicotine acetyl receptor partial agonist. It was first approved in May 2006 for smoking cessation therapy and is presently approved by CDSCO for clinical use. This drug has a long elimination half-life of approximately 24 hours. It is commercially available as film-coated tablet in strength of 0.5 mg to 1 mg, undergoes minimal metabolism, 92% is excreted unchanged through urine, achieves peak plasma concentration within 3 to 4 hours after oral administration. However, despite it is proven therapeutic effect. The tablet dosage form is associated with side effects like nausea, insomnia, abnormal dreams, headache, vomiting, and gastric discomfort, resulting in reduced patient compliance. This brings us to core problem. How can we deliver this effective drug in more acceptable and more patient-friendly manner? A major challenge in smoking cessation therapy is adherence. Many patients discontinue early due to discomfort, taste, swallowing difficulties, or delayed action. So the purpose of this research was to provide a dosage form that dissolves quickly, eliminate swallowing concern, mask bitterness, improve comfort and compliance. This lead us to concept of varicline buccal strip. The problem we are targeting is smoking has unfortunately become a trend, especially among young adults and teenagers. They often begin due to peer influence, curiosity, or the belief that smoking provides stress relief. Most tobacco products like cigarettes, cigars, vapes, or hookah contain nicotine. Nicotine is highly addictive compound that binds to nicotine cholinergic receptor present in brain neuromuscular junctions, autonomic ganglia, adrenal medulla. After administration, nicotine causes the release of dopamine, serotonin, vasopressin, beta-endorphin, and ACTH. These chemicals create a sense of pleasure, calmness, and reduce stress, which makes the brain demand them repeatedly, resulting in addiction. But the addiction is not the beginning. The real danger lies in the carcinogens generated during smoking. Cigarettes contain polycyclic aromatic hydrocarbon, tobacco-specific nitroamines, formaldehyde, benzene, aromatic amine, radioactive polonium. These substances damage DNA, disrupt cellular function, and cause mutation that lead to lung, mouth, and throat cancer. So the motivation behind this formulation is not just scientific, but also social to support the health of younger generation and reduce smoking related disease burden. A major challenge in smoking cessation therapy is adherence. Many patients discontinue early due to discomfort. So a buccal strip is designed because of its thin, flexible film placed inside the cheek. It hydrates rapidly, adheres to the buccal mucosa, and releases the drug in controlled manner. This design required careful selection of film-forming polymers, mucoadhesive agent, plasticizers, taste masking excipients, saliva stimulating agent, sweeteners, flavoring agent, and colorant. Together, these components provide mechanical strength, flexibility, adhesion, fast disintegrations, better palatability. Manufacturing methods for oral strips are solvent casting method, hot melt extrusion. Solvent casting method is more reliable for our drug varicline due to its thermolyal property. It cannot withstand high temperature. So in conclusion, the development of varicline tartrate buccal strip represents a scientifically strong and clinically meaningful innovations in oral solid dosage form. This formulation eliminates swallowing difficulty, improves comfort and convenience, may reduce gastrointestinal side effects, offer controlled release condition, enhance therapeutic acceptance and adherence. This innovation bridges the gap between drug efficiency and patient compliance, ensuring the treatment becomes as user-friendly and it is effective. Result, our formulation of buccal strip demonstrated a good mechanical strength, rapid disintegration in the buccal cavity, successful taste masking, effective mucoadhesive property, potential improvement in patient compliance. These findings suggest that varicline buccal strip are most promising alternative to tablet for smoking cessation therapy. Our formulation demonstrates that optimization in oral solid is not just a trend. It is a necessary evaluation toward patient-centric precision in drug delivery. Thank you.
I now invite the judges to ask their questions to their subsequent presenters.
Yeah, thank you. Thank you for this presentation. But we have, I think, a whole team who's working on this project. Could you see the presentation?
No.
Because, I mean, none of the slides at least were visible to me. But the
Yeah, they are not
presentation
clear.
done, I must say, was really very good. Presenter has done a good job on that front. I just have one question for you. You have developed this and you mentioned that it could also work as a control release for the drug. Did you check the dissolution stroke absorption profile of your formulation? Which is very important. Hello, we can't hear you. Hello.
I don't think they're there.
Presenters, you all are muted.
Participant number, code number, OS010. We can't hear you. Can you please turn on your mic?
The mic is on, but I think we can't hear them.
If you are not audible, please write your answers in the chat.
Or can we reserve their questions after all the presentations are over, judges? Will they rectify the problem?
Right. Yeah, we don't mind. I don't mind.
Dr. Parab and Dr. Subhashish?
Yes, sure.
So for this particular presentation, maybe if the video there were technical glitches, so maybe we'll reserve the questions after once they rectify the problem. Participants, once your technical issue is resolved, get back to the organizers. You can just do one thumbs up so that we know that you all have understood the instruction. All right. Yeah. Please come out of the meeting room. We have the next
Code number OS014. Please turn on your camera and we will be presenting your video now. Thank you.
Good morning, everyone. My topic is co-processing of ethyl cellulose and pectin ready-to-use polymer blend for targeting of drug release in colon. Co-processing can be defined as combining two or more established excipients in a specific ratio by an appropriate process. It involves preparing multifunctional excipients in a novel approach that result in physical alteration without compromising their chemical structure. Primary goal of co-processing is to produce a product with improved functionality at reduced price, which have capability to modulate the solubility, permeability, or stability. And this co-processing will address the issue of flowability, compressibility, disintegration potential. This polymer blend is prepared for colon-specific drug delivery systems to treat variety of bowel diseases like ulcer and colon cancer. This system will be capable to protect the drug from entry route. It means drug release and absorption will not occur in stomach, but the drug will absorb in colon and the drug release will occur in colon when the drug reaches the colon. Pectin offers enzymatic degradability in colon but lacks mechanical strength, and ethyl cellulose provides sustained release but not in colon responsive. This co-processing addresses the need for simple, cost-effective, and reliable polymer blend capable of delivering both BCS class 2 and BCS class 3 drugs specific to colon. The rationale behind this work was to achieve colon-specific drug delivery with controlled release characteristics with advantage of pH-dependent solubility of pectin and sustained release properties of ethyl cellulose. Drug release occur with mechanism of swelling, erosion, and diffusion mechanism. To check effect of co-processed excipient on various BCS classes like class 2 and class 3. Ethyl cellulose provide a hydrophobic barrier that controls drug release, while pectin contributes to colon-specific drug targeting through its gel-forming and enzyme-responsive characteristics to achieve the site-specific drug delivery. Here, solvent evaporation method was selected for preparation of co-processed excipients as it is cost-effective and simple method compared to other methods. Other methods are solvent evaporation method, spray drying, hot melt extrusion, and others. Preparation method, that is precipitation method for co-processing. Firstly, dissolve hydrophilic polymer. Hydrophilic polymer used is pectin. 1 gram of pectin was dissolved in 50 ml of water in beaker. Then dissolve hydrophobic polymer. Hydrophobic polymer used is ethyl cellulose. 1 gram of ethyl cellulose was dissolved in 50 ml of ethanol in another beaker. Then keep this beaker on magnetic stirrer for homogeneous mixing. Then add ethyl cellulose solution dropwise in pectin solution with vigorous mixing. The precipitation will occur and product is obtained. Then this precipitated product pour into the Petri plate and keep the Petri plate in hot air oven for drying at 50 degrees Celsius. Then scrap the product and triturate the product in mortar with the help of pestle to obtain the fine powder of co-processed excipient. Then the study was done on 2 different drugs, fexofenadine HCl, which is BCS class 3 drug, and telmisartan, which is BCS class 2 drug. Then the ratio was set like this was studied on different ratios. The ratio set was 1:0.5, 1:1, 1:2. Here, the first ratio is pectin and then ethyl cellulose. Then this batches shows the fast release, that is 8 hours, which was not sufficient sustainment. Compared the physical mixture, which released 100% within the 6 hours, where co-processed batches release 69% at 8 hours, confirming improved sustained release with co-processing. Then telmisartan, which is BCS class 2 drug, here studied with different ratios of pectin and ethyl cellulose, that is 1:0.5, 1:1, and 1:2. The results show when we compared it with physical mixture, release approximately 99.64% within the 6 hours, and co-processed batches show markedly slower release. The permeability, fexofenadine tablet permeation was approximately 52.85%. Telmisartan tablet permeation was approximately 82%. The sustained release from co-processed polymer matrices did not compromise permeability. Tablets provided control release with maintained drug transport. Data analysis. When the influence of polymer composition on drug release, this data clearly indicates as ethyl cellulose level increases from 0.51 and 2 parts. Both models, fexofenadine and telmisartan, demonstrated progressively slower release, confirming the formation of more hydrophobic diffusion limiting matrix. Improvement due to co-processing. Then improvement due to co-processing versus the physical mixture. Physical mixture dissolve almost entirely in 6 hours due to poor polymer distribution and loose structural organization. Co-processing produced homogeneous polymer dispersion, reduced particle size, better inter-polymer bindings, and lower porosity of the matrix. This modification led to the tighter gel formulation and slower water permeation, explaining to the significantly superior sustained release. Then performance of co-processed polymer across different BCS classes. Here first is fexofenadine, which is high solubility, and require a stronger hydrophobic barrier. Here co-processed polymer with pectin and ethyl cellulose ratio of 1:1 and 1:2 ratios effectively restricted the rapid dissolution. Telmisartan, which has low solubility, naturally slower dissolution, enhanced sustaining effect, showing excellent compatibility with ethyl cellulose-rich matrices. Thus, the co-processed excipient is suitable for both highly soluble and poorly soluble drugs, making it versatile for colon-targeted formulation. And then permeation behavior despite the sustained release. Here we find sustained release did not reduce permeability. Tablets maintain adequate drug flux despite slower release. Control release created stable drug concentrations, enhancing diffusion across the membrane. This supports that the co-process matrix is safe, functional, and biocompatible for colon delivery systems. Functional confirmation via characterization studies. We had studied this with FTIR, DSC, and XRD, SEM, and SEM. Results indicated there was no chemical interaction between the polymer and drug polymer. There was no peaks or degradation events, and the morphological changes confirm physical modification, only validating co-processing as a safe excipient engineering techniques. This ensures the excipient is chemically stable, pharmaceutically acceptable, and ready for future formulation applications. The conclusion behind the present work was the result found that the designed co-process excipient presents good tabletability, enhanced powder flowability, and improved sustained action. These performing functionalities were achieved accordingly to a synergistic effect obtained from a combination of both polymers, that is ethyl cellulose and pectin, and hence the formulates effective co-processing excipient. Thank you.
OS014, please on your camera and audio. Now I will request the judges to ask their questions to the respective presenters.
Good morning, ma'am. Am I audible?
Yeah, we can hear you.
Yes, good morning, ma'am.
Can we proceed with the questions? Hello?
Yeah, please, madam.
Yeah. Thank you very much for the presentation. You have done a good study and you have mentioned you are going ahead with colon targeting. Now, colon targeting requires at least 8 hours non-dissolution in the rest of the GI tract. Have you done any study on that front?
Yes, ma'am.
That your formulation does not dissolve or disintegrate for 8 hours?
Yes, ma'am.
Right. And finally, you have done some permeability studies in the gut sac.
Yes, ma'am.
Can you tell me at what pH you have done the study?
Ma'am, we had studied on pH 7.4 with non-inverted gut sac of goat.
Right.
And as it is a segment of isolated goat intestine used in as an ex vivo model for studying intestinal part for drug absorption, where the tissue was kept in a natural orientation, and we had used 2% of goat fecal content and maintained the anaerobic media for that. And we had used the lower intestinal part of goat.
Right. And within how much time could you get the complete drug release over there? I mean, none of that data was presented.
Ma'am, we had studied it for 8 hours and we had observed the results.
No, you studied for 8 hours, it should not dissolve, but then once it goes to the permeability studies in gut sac, you took 8 hours to get the complete dissolution. By that time, the drug will get excreted. I mean, since you get my point, what I'm trying to say.
No, ma'am. Can you
The
please
formulation
repeat?
takes.
Ma'am,
Sorry. Yeah.
I'm co-author of the team. So, ma'am, the complete permeation did not occur. Am I audible, ma'am?
Yes, you are auditable.
So, ma'am, the
Hello.
complete permeability study was hello,
Yeah.
hello.
Yes, we can
Then
hear
the
you.
permeability study was conducted, but the complete permeation did not occur. Like for fexofenadine, it was around 52.85%, and for about telmisartan, it was about 82%. So there were no fixed, what we can say, experimental time duration were conducted. Hello.
Some additional studies need to be done for this, supporting the sustaining effect and the permeability. Secondly, fexofenadine being class 1, I think 100% should have happened. So your polymer is retaining quite a lot of the drug. Or is it degraded? We don't know.
Yes, ma'am, we will look after it.
Yeah. So I think a lot of studies still needs to be done for this. Thank you.
I think there is a gap between what the title says and what is being spoken in the presentation and in the work. You talk about colon targeting in the topic, okay, which Madam also highlighted. Whereas whatever you're saying or the studies result in is all controlled release. So I think this basic thing has to be understood before getting into what is the objective of this entire coexcipient formation. So please look into it. Otherwise, it would lead to confusion.
Okay, sir.
Hi, thanks for the presentation. Initially, I felt that it was more theoretical because I did not see any tables or any studies shown, like Madam said. I have one question to know whether the formulation is robust. Have you done any robustness studies of this?
Presenter, did you hear the question from Judge?
No, sir.
Sir, can you please repeat the question?
I wanted to ask whether you had done any studies to check the robustness of this formulation.
No, sir, not yet. No robustness study was done. We'll do it.
Okay, thank you.
Thank you, sir.
Thank you judges, for your valuable questions. For subsequent presentation, code number 019, kindly switch on your camera. We will be presenting your video now. Thank you.
Good morning, one and all. Today we are discussing on the research topic of development and evaluation of ivacaftor liquid solid sublingual tablet to improve the solubility and dissolution profile, optimization and in vitro evaluation, presented by OS019. Now, let us discuss the background of this research. Ivacaftor, a BCS class 2 drug used in treatment of cystic fibrosis, suffer from aqueous solubility, extensive first-pass metabolism, resulting in only 20% of oral bioavailability. Liquid solid compaction offer a simple, cost-effective approach to enhance dissolution by converting a drug solution in a non-volatile solvent into a free-flowing, compressible powder that improve the wetting surface area and the molecular dispersion, Sublingual delivery further bypasses the hepatic first-pass metabolism and enables the rapid systemic absorption through the highly permeable oral mucosa. Combining the liquid-solid technology with sublingual administration therefore provide a dual strategy to address both solubility and metabolic limitation, offering a promising pathway to significantly improve the therapeutic performance of ivacaftor. Now, the rationale of the study. The ivacaftor is BCS class 2 drug with low solubility and high permeability, results in 20% of oral bioavailability due to the poor solubility and extensive first-pass metabolism. Liquid-solid compaction enhances resolution by dispersing the drug in non-volatile solvent and embedding it within the carrier and coating material, increasing the surface area, improving the wettability. Sublingual delivery bypasses the hepatic first-pass metabolism and it enables the faster and more efficient systemic absorption. Combining the liquid-solid technology with sublingual administration simultaneously addresses the solubility limitation and metabolic degradation, thereby improving overall bioavailability. Using GRAS classified carrier coating material, the liquid-solid system offers a simple, scalable, and cost-effective strategy for enhancing the performance of poorly water-soluble drug like ivacaftor. Now, this is the method of preparation of liquid-solid system. Now, first we will here see the drug solution suspension in non-volatile water miscible solvent are dispersed in hydrophilic or hydrophobic polymer for immediate and sustained release by using the coating. After that, the coating material is used. After that, we get the free-flowing and compressible powder. We can compress it into the tablet and we get a sublingual tablet and check the drug release in dissolution media. Now, we are discussing the result of the study. The pre-formulation and solid state result, FTIR showed the no drug excipient interaction. DSC and PXRD confirmed the conversion of ivacaftor from crystalline to amorphous form in liquid solid compact and supporting the solubility enhancement. SEM images showed the uniform absorption of drug onto the carrier coating material. These are the same images of the ivacaftor. And this is the physical mixture of the ivacaftor and other excipient. And these are the ivacaftor liquid solid. Third image of this is having the ivacaftor liquid solid compact mixture. The solubility optimization result, ivacaftor exhibited highest solubility in transcutal P, 150 mg per ml. The liquid solid compact showed the 18.38-fold increase in solubility over pure drug. DOE optimization identified ideal carrier coating ratio and solvent level of maximum solubility and dissolution. Tablet evaluation and dissolution result. The pre and post compression parameter remained within the pharmacopoeial limit. Optimized liquid solid sublingual tablet show a rapid drug release 51% in 7 minutes, 76% in 15 minutes, and we get 96% in 20 minutes. Pure drug tablet release only 28 to 30%. Ex vivo permeation study, liquid solid sublingual tablet permeation is 74.8% in 30 minutes. Pure drug permeation is low. 35.42% demonstrate the enhanced absorption and bypassing first-pass metabolism. Now, data analysis. The solubility and solid states result. Ivacaftor shows maximum solubility in transcutal P selected as a liquid vehicle. FTIR shows the no interaction between drug and excipient. DXC and PXRD pure drug is a crystalline and liquid solid compact converted into its amorphous form and it enhances the solubility. The same shows the uniform adsorption of drug onto the carrier and coating with the smooth amorphous morphology. Then optimization study, the significant influence of solvent level, carrier to coating ratio and polymer on the solubility and dissolution. Optimized formulation achieved 18.38-fold solubility enhancement versus pure ivacaftor. Tablet evaluation dissolution, pre and post compression parameter within limits. Good flow uniformity, friability less than 1%. Optimized liquid solid sublingual tablet show rapid drug release 51% in 7 minutes. greater than 76% in 15 minutes, greater than 96% in 20 minutes. Pure drug tablet only 28 to 30% release. Ex vivo permeation study, in that the using France diffusion cell goat sublingual mucosa, sublingual tablet was oriented in its core in contact with the mucosa. The phosphate buffer having pH 6.8, temperature we are maintaining 37 plus minus 0.2 degrees Celsius, 1 ml aliquot withdrawn from predetermined to time interval analyzed using UV at lambda max 223 nanometer. The donor compartment was filled with 2 ml of phosphate buffer at pH 6.8. The percent drug permeated for liquid solid compact was 74.8%, whereas pure drug having 35.42 42% with for the 30 minutes. This is the graph of percent drug permeated to versus time. Now conclusion. In the present study, liquid solid compact of ivacaftor was successfully prepared and evaluated. Transcutol P was used as a liquid vehicle, and Neucillin US2 with aerosol 300 as effective carrier and coating material respectively, due to their excellent flowable liquid retention potential, enhanced solubility and resolution rate obtained in the present study that indicates an increase in the bioavailability drug due to increase in wetting and surface area available for dissolution. The ivacaftor liquid solid compact exhibited 18.38-fold increase in solubility compared to the pure drug. The liquid solid compact show a superior drug release, achieving more than 96% cumulative drug release within 20 minutes. This study established a liquid solid sublingual tablet technology as robust and cost-effective strategy to improve the solubility, resolution, and ultimately therapeutic performance of poorly water-soluble drug like ivacaftor. It highlights the synergistic effect advantage of combining solubility enhancement technique with alternative route of administration to address the multifaceted formulation challenges. These are the reference of the study. Thank you.
I now invite the judges to ask the question to the respective presenter.
Thank you so much for the presentation. It was a good study that you have carried out. I had a couple of questions. You mentioned that you got more than 96% drug release in 20 minutes. And the ex vivo study that you carried out was just 74% in 30 minutes. Is this difference acceptable? And can you explain the difference?
Ma'am, 96% in a dissolution study, and got only 72% in ex vivo.
Yes, in 30 minutes. So,
yes, ma'am.
No, my question to you is this difference acceptable, or should you try to narrow down this gap? And secondly, a sublingual tablet, will it remain in the mouth for 20 to 30 minutes?
Ma'am,
I mean, just think about it. Maybe you may look at further fastening the release profile in order to achieve faster release. And what was the method, the procedure that you used for your in vitro study, drug release study? What procedure did you use? Which apparatus?
Ma'am, dissolution apparatus I have used.
Which one? Which one?
That
there are 7 disso apparatuses. Which one did you use?
Yes, ma'am. France diffusion cell we have to use in a
Hello?
in vitro. France diffusion cell we have to use in vitro.
Can't hear you. Hello?
Thanks, sir.
She's saying France diffusion.
France diffusion cell. Okay, right. Okay, thank you. That's all from my side.
And ma'am, liquid solid tablet is its permeation is 74.8% as compared to its pure drug is 35.42%. So it's a 96% drug release from 20.
Yeah, but our target should be full 100%. No? 100%, I feel we should target within less than 10 minutes
Yes.
for sublingual.
Yes.
Okay, so my question is, what is the rationale for developing a sublingual system?
Ma'am, because of Ivacaftor is a BCS class 2 drug, and it having a hepatic it goes hepatic first pass metabolism. Therefore, we are formulating in sublingual to bypass the hepatic first pass metabolism.
Okay, but in that case, as the question was raised that for 20 minutes, would you keep that in the mouth?
No,
I mean, nothing stays more than 1 or 2 minutes, and I think even 2 minutes is something more which I've talked about. So will that not
Yes.
get into the gut?
Yes, sir. We will study on that.
Yeah, thank you.
Yeah, one question that comes that the standard formulation which is approved has limited bioavailability, roughly around 20 to 30%, where it goes to the first pass mechanism. And when we are enhancing this drug release to almost more double that, do you anticipate any potential toxicity? Because this drug has a lot of toxicity effects, reported.
Yes, sir.
Because the levels in the blood will be suddenly high when you go with some. Is there a need for reducing the dose or something like that? That rationale also can be covered, You can do some search.
Yes. Yes, sir. We will study.
Thank you so much.
Thank you, judges, for your valuable questions. As code number 011 is now available now, kindly switch on your camera. Judges will be asking their respective questions.
Hello. Good afternoon, all. Am I audible now?
Yes.
Sorry for the inconvenience. Hello.
Judges, you would want
Am
to
I
have
audible now?
this presentation played again just to have a quick recap. Ajab, madam?
Actually, the presentation was not visible at all, so doesn't make a difference.
So I have a question related to your I think you had mentioned about buccal strips, right?
Yeah.
So since we couldn't understand or see the presentation, could you give a clarity on the rationale for using this molecule? And at the same time, since you have studied that, since the dissolution is from the buccal cavity, have you done a pH sensitivity test whether this particular molecule is stable in that pH or not?
So our main motive for this topic was better palatability and convenience for the patients or for the public. Right now, we have tablets of this varicline tartrate in the market, which has many adverse effects like nausea and gastrointestinal irritation and all. So the main motive was to decrease that, reduce that adverse effects. And we had not done practical test on this because right now we are second year pharmacy student, bachelors. So we will work on this in detail and we will keep doing research on this. Not only theoretically, but also practically in future.
Sure. So once you develop a product which is for a particular route of administration, make sure that the stability is established, in that particular pH.
Yes, sir.
Okay, and at the same time, I think you picked up some information from the other presentations as well, that when you are giving it on a buccal system, then it has to be palatable in terms of, the taste also makes a lot of difference. So it's
Yeah.
not just about the release, but also there are certain organoleptic characters which needs to be taken care of.
Yes, sir.
Yeah, okay. Thank you.
Thank you.
Hi, thanks for the presentation. Although I was not able to read it, I want to know what excipients you had used in this strip.
So excipients like taste masking agents, flavoring agents,
The main film former, what
saliva
was the main
stimulating
film?
agents.
What was the main film former? Have you used starch there? Any
Right
starches?
now, we had not done any practical research on this.
Okay.
We can use polyvinyl thing for film forming.
Actually,
We will do
this product
practical
has an
research.
issue. It was reported having a NDMA, and there was a recall for having a potential carcinogen because it is incompatible with excipient like povidone. So when you make a buccal strip, I wanted to understand whether have you done that study, to understand the incompatibility, not only the excipient, also stability in the pH, like you said. Okay, so and the method you are proposing was hot melt, correct?
Yes, yes. No, no, no.
Yeah.
It was not hot melt.
Casting.
Yeah, casting method. So
Okay, okay. Thank you.
Yeah.
thank you. sir.
So we understand this is just a theoretical project that you have given a proposal for. You must have also researched how you will carry out the dissolution profile or the absorption profile of this particular formulation. So any ideas on that?
After formulation, ma'am, we can do dissolution test in the pH of saliva. We can make in vitro type something for dissolution test, which is as similar as pH of our saliva.
Mm-hmm. Right.
And we can
you,
test that.
any particular disso profile in mind, release profile? Because see, since you have not started the project, it would be good to design your formulation keeping the endpoint in mind. So you decide what the disso profile that you would like to achieve upfront and use that as your critical quality parameter and do it by DOE, design of experiments, QBD, and achieve it. No? This is what my advice would be since you have not started the project already.
Okay,
Yeah,
ma'am.
thank you.
We'll keep that in mind. Thank you, ma'am.
Yeah, I don't think we have any other questions.
Thank you, judges, for your valuable questions.
Now we request the judges to share a few words about their experience on today's events. Dr. Parizad, please can you start?
Yeah, thank you very much. I would say it was a privilege to listen to these young researchers. They have given it a lot of thought and a lot of input. Some places we did find the projects a bit lacking, for which excuse us if we have been quite blunt in providing our feedback, but we really want you to take it on board, work upon it, and come up with projects which are almost up to completion stage, where we can really evaluate what you have achieved. Just one point to our presenter 004. He is planning to patent his research. My dear, if you are planning to patent something, you don't present it first. So you first apply for a provisional patent and then go ahead and put it in public domain, because there are chances that the patent may not be granted. So this is for everyone who's attending. Just keep it in mind. If you have a novel idea, just go ahead and take a provisional patent on that idea and then proceed to work on it. so that no one else will have the opportunity to copy it and avoid your patent from being granted. So thank you very much to each and every one of you all. Whoever wins, wins, but all of you all have done a good job in presenting to us today. So thank you.
Thank you, Dr. Paridad, for your insightful view. Now I request Dr. Subhashish Chakraborty to share their view.
Okay, so great job, guys. I think there was some thought-provoking stuff that I could hear. But at the same time, I recall one of my conversation with my guide long back during my PhD when he said that 70% of your work is done when you have finalized the title of your project. Okay, so it looks quite a normal statement, but there is a depth to that. Okay, the reason being that once you know what you're going to do, okay, the work becomes easier. And that is what I'm trying to stress here, that thinking about the project, questioning yourself, questioning the rationale behind the execution of the project, looking forward for the right method and evaluation so that you know exactly what you want from the project, and accordingly then take a, it will become easier for you to find a conclusion to it. So this entire holistic way of looking towards the problem statement till a conclusion is something which needs to be seriously taken into account. And that is what I'm trying to convey a message to each and everyone, that please be clear about the problem statement, followed by what exactly you want to achieve, which is called as the objective of the project. And I think once that is, these two things are clear, you will for sure get a clarity of how to execute it. And once you get the results, then analyzing the results itself, is a, I would say it's a fun activity, because you'll enjoy getting the data and deducing. Give me a moment. Okay, and then finally giving a meaning to this entire work that you have done. Okay, so I think the one word which comes out is thinking. And as madam also mentioned, I think there was a thought involved, thinking involved, but I think it has to be more concrete, more structured, okay, and to a better depth. Okay, so thank you very much for presenting. I think it was a pleasure listening to all of you. Whoever wins is okay, but I think you each and everyone did a good job. Thank you.
Thank you so much, Dr. Chakraborty, for your insights. Now, can we please, Dr. Rajesh Parab, share your views?
Hello, everyone. First of all, thank you for inviting me to be the judge for these excellent presentations. Like Subhashish and Madam said, everyone has done excellent job, and it was an opportunity for us to understand what's going on and how the thought process is happening at your level, at the college level. And I know there were some undergraduate, some postgraduate students presenting here. Excellent ideas, very interesting topics. I know there's always a way to improve, and your guides will help you to improve it, following a certain methodology. When you do a research methodology, you do a literature survey, you do a proper planning, you may then decide on the title and then work on end goals. And while doing that, there are a lot of parallel other activities that you have to understand. Is there any other similar work done? Also, keep in mind the regulatory framework, the surrounding that active, any adverse reportings of that active, so which will help you to make the study a complete study when you present it. So congratulations to each and everyone. Even if we have this limitations of selecting few winners, but getting to this platform itself was an achievement, that you guys have done a work which was worth presenting to this forum. Thank you again.
On behalf of the entire organizing team of the 6th SRC, it is our honor to present the vote of thanks. Our sincere gratitude to our distinguished judges for their time, expertise, and insightful evaluations. Your thoughtful guidance has enriched this session immensely.
A heartfelt appreciation to all our participants. Your passion, dedication, and innovative thinking have truly elevated this event. You are the driving force behind this competition, and we applaud your outstanding efforts. Lastly, we extend our warm thanks to our audience, faculty members, organizers, and technical team. Your support and enthusiasm have played a vital role in making this event a resounding success. Thank you all, and wishing each one of you continued excellence and growth in your research endeavors.
Thank you, Dr. Parizad, Dr. Rajesh, and Dr. Subhashish.
You're welcome.
Thank you. So what is next, Team BNCP, if you can just quickly tell us?
We now look forward to the awards and valedictory session, where we will recognize and celebrate the exceptional contributions and achievements of our participants. The session will begin after a short break. I request all the participants to join us tentatively at 2 p.m. for the valedictory session.
Just
What time?
just hold on. Just hold on.
It will be around 2 p.m.
Any comments from the judges to the participants with respect to how do they conduct the research, or what you felt about the oral hackathon, because this was our new endeavor. So if you can share your comments about what ideas to be inculcated, that would be really great, madam.
We already shared, Sujata. Hello.
Okay. Any concluding remarks from other two?
We have already done it, ma'am.
No, both of them, all three of them, they gave their comments. Yeah. So what time we are meeting again, Sujata madam?
2 o'clock, because I think we are done with oral hackathon, but PhD and
the
hackathon
AI is ongoing. AI in healthcare is ongoing, and PhD is still ongoing. So, Dr. Afrin, what time we should be informing, because we request ACG team to be there at valedictory when we announce the winners. And any one of you, we would request, we will share the winner's name. So any one of you would be declaring the winner's name.
Thank you, judges, for your insights in this and judging the participants. So because all the other sessions are currently ongoing, like Dr. Moomin said, we would specifically call you once the score chart is ready, and we will also let you know by calling to join us for the valedictory function.
How do we share our scores? Just I had a question. How do we share our scores? We shared that list which you have sent us.
Yeah, you can share it with
by email. Send it by email.
Yes, send it by email. Put your signature on that scorecard and share it with us. Share by email quickly, so that we'll compile the result, and we will inform you. We will share the winners list, and you will be then declaring 1, 2, 3.
Are the judges allowed to discuss together and share the score?
Yes. Yes. Yeah. So, Afrin, do you want to create a room?
No, we'll
so that only 3.
We'll contact on WhatsApp. Is that okay with Subhashish and Rajesh?
Yeah,
we'll do a WhatsApp call and
And ma'am, I may not be able to join the afternoon session. I have some other commitments, so I thought we will be done by 1 o'clock. So I had taken up some other commitments.
Okay, no problem. But we would request at least one of you or two of you to remain present during valedictory, because you will be announcing the winners.
Okay, so Subhashish, you will be there?
Yeah, I can be there.
Yes. So both of us will be there, and we will announce, we'll distribute.
So what I will do is, so Parizad madam, you will be announcing solid orals, and Subhashish sir, you will be announcing AI in healthcare. We will share the winners.
Okay, done.
All right. So you can have among yourself discussion, and we'll then get back to you. We will again share the inaugural valedictory link once again, just for your quick reference, so that you can join immediately.
Thank you so much. Thank you.
So thank you very much.
Thank you. Thanks
Bye-bye.
everyone.
Yeah. See you at
Bye.
valedictory.
Bye-bye.
Thank you, Dr. Rajesh.
Yeah.
Yeah. Thank you. Bye.